Transfer any appropriate amount of sterile water, magnesium chloride, 10x reaction buffer, DNTP, forward primer, reverse primer, and tack DNA polymerase to a PCR tube with a micropipette. Mix the reagents in the PCR tube using a vortexer. Centrifuge the reaction mixture in the PCR tube using a mini-centrifuge to collect all drops from the inner wall of the tube. Preheat the thermal cycler to 95 degrees Celsius. Transfer 40 microliters of master mix into PCR tubes A, B, and C. Add 10 microliters of DNA template control DNA and sterile water into PCR tubes A, B, and C respectively. Mix the reagents in each PCR tube using a vortexer. Centrifuge the reaction mixture in each PCR tube using a mini centrifuge. Place all the PCR tubes into the thermal cycler immediately. Push the tubes down completely into the wells and close the lid of the thermal cycler. Start the PCR program according to this thermal cycle. Place the PCR tubes on ice after the reaction. Using gel electrophoresis, we can observe the DNA fragments amplified in the polymerase chain reaction. Perform gel electrophoresis using the reaction mixture in PCR tubes A, B, and C. Observe any DNA bands formed in the gel. Record the results.